Perfect. Okay, over to you, Don. Thank you very much.
Great.
We'll do about 40 minutes and then Q&A and then some feedback from Carl afterwards.
Sounds great. Sounds great. Thanks very much. Today I'm going to, in many ways, continue on from Fan Lu's talk last week. And I'm going to bring us to a new paper that we've submitted with this title, emphasizing excitatory-inhibitory resonance, looking at the interaction between excitatory and inhibitory mechanisms, specifically across the layers of the cerebral cortex. We'll start with our evolutionary mantra that we bring to our theoretical work, in which we look at cognition as a continuation of neural development. And in fact, it is neural development, that the mechanisms of the brain are developmental. And as such, we will try to understand the evolution of structures as well as the ontogenesis or individual organization of structures as a clue to their function. And this is an approach that Fan Lu and I have developed over a number of years, where if we look at the structures of the brain, seeing how they develop gives us important clues to their current function. And in this framework, we're going to be looking at excitatory and inhibitory mechanisms, not just at the local circuit level, but at the organismic level. And this is an effort we started a few years ago. And this paper in developmental psychobiology looked at embryogenesis and specifically the organization of sleep in the embryo as a clue to how excitatory mechanisms, particularly active or REM sleep, and inhibitory mechanisms, particularly quiet or non-REM sleep, become critical for the self-organization of the brain differentially. And we were quite interested in the genetic or genomic components of the developmental process, where the modern evidence is giving us important clues to the organization of the brain. And this is key to Fan's talk last week, which I will build upon. And the prosomeric model developed by Puelis and Associates. And the core principle is that the organization of the neural tube, which actually stops in the hypothalamus, remains important to the organization of the telencephalon. And we describe this as vertical integration, the way that the telencephalic structures that we see as the cerebral hemispheres depend upon fundamental excitatory mechanisms that evolved along the dorsal trend and inhibitory mechanisms that evolved along the ventral trend. Fundamentally, the excitatory are involved in sensory integration and the inhibitory and motor control. And as we look at the morphogens that differentiate the neural tube, as Fan explained, we find that certain morphogens controlling the fate of neurons from the roof plate, primarily organizing sensory functions, differentiate excitatory neurons. Glutamatergic neurons depend upon these morphogens for their differentiation from stem cells. Conversely, the floor plate differentiates GABAergic neurons under SHH control. And we see this in the neural tube up to the hypothalamus. And then we see the telencephalon developing from HP1 out of the hypothalamus. And at that point, we still see the excitatory inhibitory mechanisms, but in a more complex fashion. And as Fan explained, this leads to what we describe as the telencephalic rotation, where instead of just dorsal and ventral, we now have SHH acting in the anterior or rostral divisions and setting up a rostral to caudal division of the brain that we recognize in mammalian brains now. But fundamentally, this dorsal ventral distinction is an excitatory-inhibitory one. So here we have the tiger salamander in Herrick's classical anatomical analysis. And what we find is the dorsal division, the pallium, the primitive pallium, is excitatory with glutamatergic dominance of the neural activity. Conversely, the ventral neural tube, which is the basal ganglia and subpallial structures, is inhibitory. Now, as we get to mammals, this organization is going to be more complicated, but we see the excitatory-inhibitory division as a clue to vertebrate organization generally, and we'll extend it to mammals in the dorsal and ventral divisions. And the connections with subcortical areas are also more simple in the tiger salamander and other amphibians. Particularly important is the continuity of the forebrain with the thalamic systems, the lemnothalamic system, which is connected not to the midbrain, but down to the rhombencephalic, the pontine level of the reticular activating system. That lemnothalamic pathway connects those lower brainstem with the thalamic regulation up to the pallium, exclusively to the pallium, the medial and dorsal divisions of the pallium. The colothalamic pathway that connects the midbrain, but not the lower brainstem, is specifically targeting the striatum. And this is an organization that is important as we go to higher levels and the more complex dominantly excitatory-inhibitory systems of the interdigitated mammalian brain. And I think if there's one principle of the telencephalon in mammals, it's this interdigitation of excitatory and inhibitory mechanisms. But we think the primordial roots still shows that we can separate the excitatory feed-forward qualities of the dorsal division at an organismic level from the inhibitory feedback control that's most connected with the ventral divisions in mammals. And here we see a corresponding complexity of the subcortical projections, where the lemnothalamic system and the colothalamic system share connections with the striatum and the dorsal pallium. But nonetheless, we argue there is still an excitatory and inhibitory organismic function. Now, in the mammalian brain, as Fawn explained, we find invasion of the three-layered pallium by inhibitory neurons, GABAergic neurons that migrate out of the basal ganglia and ganglionic eminences in embryogenesis to take up intermediate positions and create the six-layered cortex. So there's no doubt that excitatory and inhibitory mechanisms are closely linked in the mammalian brain, and that gives new capacities of cognitive and behavioral function. But understanding their separation continues to be important. So in our analysis, if we fold the neocortex out to its major pathways, we find that the cortical hierarchy has its top or executive level in the limbic system, whereas the more eulaminar or differentiated six-layer cortex is out toward the sensory and motor divisions. And within this hierarchy, there is a feed-forward pathway, a predictive pathway in active inference that is excitatory, emerges from layers 5, 6 up to superficial layers. In the model of Bastos et al. That we'll talk about a little later, this is the predictive pathway, and it's fundamentally excitatory with certain neural oscillations, such as theta oscillations, that carry this predictive information and the mechanisms of active inference. Conversely, the opposite pathway from sensory data provides constraints and, in fact, error correction for these predictive excitatory. And these are indeed inhibitory, and they're restricted to superficial layers, and they're more focally differentiated compared to the global excitatory oscillation patterns. And in the neuroscience literature, most people think of the input pathway as the feed-forward pathway. They talk about it that way. But we're going to say that's wrong. It's really feedback control. And in fact, in active inference, the sensory constraints are feedback control in the cybernetic sense. And similarly, the pathway from limbic, which we call limbifugal, is feed forward, not the term of feedback, which you'll find in the literature. So we have a small problem of being misunderstood by the terminology of neuroscience, and we've thought about trying to change the terminology, but it really doesn't work. And over a few papers, let me kind of briefly emphasize how we've seen these feed-forward and feedback pathways throughout the brain. So in this paper in Cerebral Cortex N23, we emphasized that the dorsal attention system, which has the limbic base of the default mode network, is seen as the endogenous attention system, where internal organismic control leads to the regulation of attention. That's associated with this dorsal pathway, which we argue is fundamentally excitatory in its primary mode of operation. Now, we have feed-forward and feedback control in both neocortical pathways, both the dorsal and the ventral. But there is this overall bias where the ventral pathway has a feedback bias, and it's supported by the extensive agranular layer that's more developed in the ventral division than in the dorsal division. So in fact, we see a kind of a primordial emphasis of the three-layer 1, 5, and 6 in the dorsal pathway, with kind of a minimal layer 4, particularly in frontal regions, compared to the feedback dominance of the ventral attention system, so-called exogenous attention, and with its underlying support by the ventral limbic extended amygdala areas across the brain. So these higher level functional divisions reflect a fundamental anatomical difference that was recognized by Sinides many years ago and is captured by this nice anatomical description by Giacchio, where we find that the dorsal limbic control extends through the dorsal neocortex, shown in white here, emanating from the hippocampus and the cingulate cortex, compared to the ventral limbic from the periamygdalar regions and anterior temporal orbital frontal areas, here shown in gray. So the qualities that emerge here have a limbic base. That's the essential idea that we're arguing. And this limbic base has an affective element and an affective bias that is coherent with the excitatory nature of the dorsal pathway. In fact, we argue this is regulated by phasic arousal. And this excitation leads to a dimension of affective control that is from depression to elation. Greater excitation is greater elation, and it's a feed-forward regulatory bias. Compared to tonic activation, that fundamentally we argue has a quality of inhibitory control. But in both cases, the modulatory properties, the cybernetic properties, are complex. The excitation cannot run rampant because it leads to seizures. So instead, the elation mechanisms of the dorsal pathway have an inherent habituation bias mediated by norepinephrine. And the inhibition doesn't just stop neural activity, it leads it to be constrained and maintained in time with a sensitization bias. So these regulatory properties are complex and not necessarily intuitive at first, but they're fundamentally the affective, the emotional controls over the activation and arousal of the brain. The place where this theoretical model, I think, became most interesting to me was when we recognized the roots in sleep and the excitatory and inhibitory roles of the memory consolidation that occurs in sleep. And so we tried to put this together in a paper earlier this year with the following alignment that illustrates different neural mechanisms for different memory systems. In fact, we've really argued that these sleep stages show us underlying memory systems. And the non-REM sleep that leads to explicit memory, we think is primarily under inhibitory control. Now, the whole brain is asleep here. It's a state when the entire brain is modulated, but the regulatory mechanisms come out of subpallial and but very close to ventral limbic control. So the claustrum regulates slow oscillations and big inhibitory downstates across the cortex. That synchronization of cortical activity is critical not only to the homeostatic regulation of the synapses, but it nests the thalamic reticular nucleus spindles. And the sleep spindles then are a second inhibitory mechanism, and those nest or organize the hippocampal sharp wave ripples. So this entire mechanism is critical for consolidating explicit memory, which we think are the unpredicted events of the day. The things that have implications for corrective reorganization of neural assemblies in the continuing process of active inference in sleep. Conversely, we have REM sleep, which is fundamentally excitatory with the limbifugal control. And here there is exclusive excitatory organization of the neural architecture with cholinergic control. And we tried to look at a way to understand the information organization in this. And we pulled on Hinton's sleep-wake model of generative exercise. So Hinton and associates showed they could stabilize the priors of a connectionist network by turning off the criteria for it to meet input and just running the generative model loose over a period of time. And what happened was it made for a more simple and effective prediction, more simple and effective generative model, as the generative model kind of organized itself in the absence of corrective feedback. Somewhat unintuitive at first, but we think this actually might be a kind of a computational insight into the way that the excitatory cholinergic regulation of REM sleep produces our dreams, but at the same time has the reorganizing function. We call it reconsolidation in REM sleep, that after this non-REM disruption with new information, we need to reorganize our Bayesian priors. And that's indeed what we do in our dreams. So we see this as active inference continued during sleep, but separated. Instead of being simultaneous, it's now alternating. And the interesting clue is, and this really is kind of integral to Fahn's talk, is that the organization of these mechanisms in the telencephalon comes from the hypothalamus. So even whereas we've argued over a few papers that the limbic system is the executive control, the top of the cortical hierarchy, not only in an anatomical, but in a functional way, now we're really pushing this down and understanding that the top of the hierarchy is the hypothalamus that is regulating the limbic system, and indeed these two stages of sleep, where the hypothalamus is regulating non-REM sleep, and while that's happening, it's leading to the suppression of autonomic function in the parasympathetic dominance that's observed in non-REM sleep. Conversely, in REM sleep, we see these surges or storms of sympathetic activation, excitatory autonomic control during REM sleep. And we think these visceral processes, the autonomic processes, are entirely consistent with the hypothalamus and its regulation of ventral and dorsal limbic divisions, respectively. In this paper, also this year, we tried to put these together and argue that the excitatory-inhibitory balance in waking is a opponent process, that both the non-REM and the REM are active, not necessarily exactly at the same time, but in a kind of a dialectic of excitatory-inhibitory balance. And in fact, as we look at models for how that could occur, there's very interesting suggestions that the brain is a system that becomes most functional at criticality, at the point of exact balance of excitatory and inhibitory function. And yet it's not exact all the time, that in order to build the feedback control from the non-REM memory system, we have to have dominance of feedback control at certain times. At other times, we need feed-forward control. We need the excitatory to help make predictions, see the big picture. And so there's what we call the variational criticality, something of a contradiction in terms, but that provides really almost like a variational Bayesian process where we can make predictions, use the integration of priors at certain points in the cognitive process, and look at the posterior distribution and the feedback constraint at other times. And so obviously, waking cognition, even though we say that consciousness emerges at the criticality of balance, depends upon the sleep mechanisms that pre-organize these for daily waking functionings. And we'll return to the way the subcortical systems regulate this criticality. An idea we're trying to understand right now is how criticality goes all the way down through the subcortical foundations. So in many ways, that is preparation. And what I want to emphasize today is a model in which we look at the resonance of excitatory-inhibitory mechanisms quite literally, that as the excitatory feed-forward waves come, such as from theta oscillations, with strong origins in 5-6 in each cross-regional mechanism. So limbic at the left, heteromodal for large areas of neocortex, unimodal association, and finally primary and sensory regions, that these theta oscillations are linking these functional brain systems in an excitatory way, and they meet the superficial oscillations, the inhibitory oscillations. Primarily, these are gamma oscillations that are the storage of working memory, but also regulated by beta. And here's where we see the basal ganglia and subpallial control coming into the inhibitory GABAergic influences on these superficial layers. But the superficial control is fundamentally feedback in an information sense. It's limbipedal, coming from the outside to the limbic, compared to the limbifugal coming from limbic toward the outside. What we argue is that when these meet, there's not only phase amplitude coupling, although we'll emphasize that and talk about it, but there is an actual resonance, an end-to-end matching. So it's not just that the gamma are nested within theta, they are exactly nested at the point of a resonance. And when the resonance occurs, it changes both operations, that the traveling theta waves meet these more local gamma waves and they resonate. And that resonance causes the activation to persist over time. Whereas the traveling waves are traveling across these territories, interacting in important ways. It's when they resonate that we can create early LTP. So the excitatory-inhibitory resonance is a mechanism of memory. And we think it's a mechanism of synaptic EI resonance that can contribute to the computational process of active inference. That excitatory-inhibitory resonance is the memory mechanism. So the model we've built on for many years is microcircuits in predictive coding. And here the terminology is opposite from what we're using. The forward connections from sensory, we think, are really the feedback connections, providing constraint for active inference. And the backward connections are not backward, they're forward, because the limbic system is the base of generative predictions. But nonetheless, the model we have here argues that active inference can be computed by the microcolumns, the microcircuit. And in fact, it's the translaminar resonance between deep and superficial layers, not just within a column, although it is that, but primarily regulated across columns in the process of active inference. There are good neurophysiologic mechanisms for theta gamma coupling, and we're trying to build upon those so that this speculative theoretical model can be looked at in relation to neurophysiology. So theta gamma coupling has been a very interesting model for a neural code for a computational process, essentially. And the Lissman and Jensen model we found particularly nice for illustrating the very clear theta gamma coupling that occurs in the hippocampus. And here we find spatial cells show a strong modulation by theta rhythms, but they also show the gamma oscillations and single unit firing. So over on the right, we have from the Lissman and Jensen model, cells that are within the place of that are marking the place cells. So when the animal is in the physical position marked by the place cells, the neural activity is very high. And indeed, it's modulated by the theta rhythm. Whereas when the animal's out of the place, the neural activity is lower and the theta modulation is lower. When they look at in this experiment, they looked at the phase of theta, they find that the phase precesses and aligns itself with the animal's position in space, consistent not only with the place cell coding space, but the global organization of the place cells by the theta rhythm. And somewhat similar mechanisms can be seen in the human brain, primarily with intracranial recordings. So Canolti and Knight have shown here is a slower theta rhythm, and it groups and organizes the higher frequency gamma. So here we find 40 hertz gamma, the low gamma, and then the higher 100, 120 hertz gamma. Both align with the theta rhythm and are organized by theta. The particular low frequency may vary. So here we have theta again as the primordial one, organizing the gamma rhythms and the phase of theta determining the power of gamma. But here it is in alpha rhythms, and we see this very nice phase amplitude coupling of alpha. So our argument is that this is not simply a modulation of the higher frequency gamma, that at critical points, there is a resonance and the waves stop and maintain their coherence through that resonance over the region that then becomes the long-term potentiation, the early long-term potentiation. So the argument is within this global excitatory-inhibitory landscape, we find these local events where the very tight coupling of excitatory-inhibitory influences is necessary for memory formation. So that's the basic model. And what I'm going to spend just a couple of minutes talking about how to test it, because I think it's eminently testable. And we have the tools to test it. We've been working for years to develop these tools. And if only we had adequate funding, we will indeed do the testing for it. An interesting element of what we recognize is that we can examine cortical activity not just with the EEG from our high density arrays, but also from the impedance. Electrical impedance tomography examines the activity of the brain and the conductive properties of the head by injecting current at a small amplitude, but a precise frequency, and then demodulating that current from multiple other areas and looking at the voltage potentials that are created from the injected current. That gives us an idea of tissue properties that includes not only the kind of general conductivity of the brain that we need for source localization, but neural activity. That neural activity changes the impedance of brain tissue. So here are some classic findings by Ross 80 a number of years ago, back in the early 70s, I think, where he would record the impedance. So the measure here is not voltage, but ohms, the resistance of or impedance of the tissue. And when the animal would approach food presented, there would be a big drop in impedance and an increase in extracellular space as a result of the activation of regions. And in fact, this was often the greatest around the hippocampus, suggesting there were resonant states, oscillatory resonance that create these impedance drops. And then there would be a rebound in impedance, maybe from glial reactivity, or we don't really know what, but a very slow impedance domain compared to the very quick electrophysiology and neural activity seen by dipolar voltage fields. So we are now fusing these two modalities for source localization. So for many years, we've used electrical impedance to build our head model, to understand the conductivity of each form of tissue that blocks how the brain activity propagates to the head surface with the bone of the skull as the major impedance factor. But our source localization, we're now adapting to include the brain impedance, the small activity of the brain, and we're using the same analytical techniques. So we found that the Bayesian multiple sparse prior inverse operation from Friston and Associates 2008 is an excellent way for EEG source localization and greatly improved over other inverses. And we're now trying to apply it in a similar way with similar simple constraints of smoothness on the cortical surface for the impedance and doing joint recording. So the idea is that we look at a task in which we know there is memory, and the memory varies. So in a paired associates verbal learning task that we're using to look at the effect of sleep on memory, there are times when you remember the correct word and you can generate it, other times when you cannot remember it and you fail to generate it. And our argument is there will be excitatory-inhibitory resonance during this recall period. It'll be flagged by theta gamma and perhaps other oscillatory coupling with a resonance that will mark the key LTP, early long-term potentiation elements, and we'll see it in the joint EEG and impedance as we source localize it. So that's essentially the goal for testing. We extract the cortical surface from the individual, we tessellate it with patches, and in this patch, we'll simultaneously solve for the voltage field and for the impedance of that cortical region. Now, we know we have some clues that when there's activity in deep cortical layers, there's a positivity created at the head surface. And if there's an activity or sink at the superficial layer, there's a negativity at the surface. And so, in fact, these cortical layer processes, these laminar features, are generating the slow cortical fields we see in the event-related potential. So here's a 128 channel array. We're looking down on the top of the head. The nose is at the top, the posterior in the back. And what we find is that there are certain features like late positive complexes that are positivities at the head surface. Those are often recognition or confirmation of the cognitive process. In contrast, when there is negative feedback and the word or the event is unpredicted and associated with a high degree of uncertainty, we find the opposite, a surface negativity, described as the N400. So there are voltage fields that give us good senses, and our effort now will be to search for not just the broad event-related potentials, but the tight and local oscillations that we think will be capturing the neural activity, the translaminar resonance of the cortical columns that are capturing this process of active inference. So here we're illustrating with Wright and Bork's model the limbifugal excitatory pathways in red from paralimbic out toward neocortical regions, and the reverse direction of inhibitory control shown in blue. So that's essentially the model that we have, and I think that's the end of my talk. So I'll quit there.
Great. Thank you very much, Dom. That was super interesting. Okay, so now we have some time for questions. If anyone has a question right now, please raise your hand. Andreas, do you want to kick off?
Yes, thank you for a wonderful talk. It's just so much exciting, not inhibiting, but exciting. I like the idea of a dialogue, and I'm wondering if I understand that dialogue correctly. I've been collecting hundreds of examples of dialogue between an unconscious answering mind that knows and a consciously not knowing questioning mind, and then a third mind that kind of balances the two, makes sure that they're saying the same information in different forms, and then chooses which one to proceed with. And so I think this is interesting for me, of course, but it's interesting that you're saying that the inhibitory mind is the sensory mind, like maybe it's giving constraints, and you're saying that the active conceptual, I guess, mind is the excitatory mind. And so that I think is quite, so that really seems to say that creatures are living from the inside out.
Yes.
And then I'm thinking...
so you've seen Busaki takes that literally. He says the brain works inside out. It's not that the sensory world determines the brain and the mind, but
Hmm.
the brain has its own endogenous inside-out projections. And I think that's what we're saying. We're saying that the excitatory process is it's the other side of self-evidencing. It's manifesting into the world in a projectional sense. Yes.
And that from the very beginning, based on the developmental study, well, how the brain evolves. You're giving evidence, I think, for a third investigatory mind that balances this answering mind and questioning mind. And that would be, I always thought it was in the basal ganglia, perhaps, but you're saying it would be it's in the in this limbic lobe in the hypothalamus, or how do you picture that balancing?
The balance, I think, is the organismic integration. Uh, and it can occur at different levels, but fundamentally, we would say there is an excitatory process and an inhibitory process. Only two, and those two give rise to emergent properties that you might be imagining as the
Mhm.
interrogating mind or the integrating mind. So, what we say is that integration, you can't find it any one spot because
Mhm.
at each level, it's an excitatory and inhibitory exchange.
So
Um,
you're not finding or you're not seeing this third mind, so to speak, but just based on introspection, like I can see things coming at me unconsciously, and then I can interrogate them consciously. But I also see that I can put aside all of my experiences, I can put aside all my questions and preconceptions, and I'm left in some kind of void. And in that void, there's very basic frameworks that connect the possible things, like levels of knowledge, like whether, what, how, why, or like the debate between free will and fate, or like the learning cycle, the scientific method, taking a stand, following through, reflecting. Like there's a whole conceptual framework world that seems just latent there. I think Michael Levin talks about the Platonic space. So there's a Platonic space for putting this together. Now, you might not see that. Another place, and in biology, the history of biology, there's many places, but like the left hemisphere and the right hemisphere. So implementations can be of all sorts, but the idea, if you look at the user requirements, which you've basically led to, user requirements insist that we should have a mind that knows answers and a mind that doesn't know but asks questions. And so, but if one only looks at implementation, they would never find that. But if you think about it, like that's, I mean, why would you say we have two hemispheres?
well, that's another talk, but fundamentally, there's a lot of evidence that the dorsal pathway is elaborated within the right hemisphere, and that the ventral pathway is more articulated in the left hemisphere. And in fact, left hemisphere language functions require the kind of inhibitory structure that the ventral division gives to its object pathway specifically. Um, Similarly, the right hemisphere elaborates the projectional feed forward qualities of the dorsal pathway. So I think laterality is overlaid on these systems. And in fact, the laterality of the dorsal attention system and the ventral attention system are interestingly asymmetric in the attention literature. So I think the left and right has a more fundamental basis, and that is the dorsal ventral divisions.
And then the notion of consciousness, like as a higher not conscious experience, but cognizance, let's say, as a higher a higher level of awareness, I would say a third level of awareness, that would be in neither hemisphere, I think, focused. That would be, I mean, that would be in the thalamus, right? Or in the base. And it would be.
Or no, it might emerge dialectically
Hmm.
from the opponent process of the left and right hemisphere. And in fact, I think that the models of cognition from classical phenomenology
Hmm.
um, point to the process of cognition from Kant to Fichte and then on through many of the other philosophers
Mm-hmm.
and modern phenomenologists, argue that consciousness and the integrated cognition is indeed dialectical. It emerges from a holistic beginning, a thesis, and a contradictory and inhibitory feedback mechanism, antithesis. And so the synthesis of cognition is not separate from this opponent mechanism. And
You
I'm
don't think
indeed,
you don't think it helps to have a third switch that kind of
no,
okay, so we disagree. But it's very helpful.
that's right.
I'll write you with more. But I just want to thank you for your talk. It's just very wonderful. You're opening up this world. Thank you.
Good. Well, I'm very interested in your arguments here for the third level, and I'm interested to argue with you about it. And
Yes, okay. That's
so thank
fantastic.
you,
Thank you.
thank you very much.
Thanks, Andreas. If there's no more questions for the moment, perhaps I can ask. Going back to the working memory task, which you were describing at the end, is there do you have something specific in mind with regards to the generative model which is being implemented in these pathways? Which aspects? Is it the posteriors which are being computed over time, or the priors which are being transmitted, which links to the working memory which you're trying to pin down?
Very good question. Very good question. And this is what we're trying to think about right now. And I can kind of reiterate the model and how the model would fit with it. And that is to say that the working memory, let's say you see the target word or the stimulus word, and you're trying to generate the target. You're trying to bring up the memory and recall it. Fundamentally, that's the excitatory process. That within your connectionist pathways, that word, that memory, is latent, and you're trying to articulate it. So the source is limbic and the generative excitatory pathways, but the recognition is the inhibitory process. So it's when these meet together and create this translaminar resonance that you have the recognition. So I would say that you begin with the feed forward generative process, but it must match and resonate with the feedback corrective process to have that moment of memory. And indeed, I think we will see it as a pattern of resonance in the cerebral cortex.
Right. You also mentioned Hinton's model at one point.
Yes.
And that one's interesting because it explicitly has a bottom-up recognition pathway, which is different from the regular, what you would call feed forward, but other people might call feedback from the generative model. So
Right.
Normally in predictive coding, we just think of having a top-down generative model, and it's the errors which are being propagated upwards. But in that particular Helmholtz machine, there's a second network which during the sleep phases, when the generative model is generating these fantasies, it's actually training this bottom-up model so that in the wake phases, the bottom-up model can actually bypass the need for doing sort of feedback and just map data directly onto posteriors going upwards. And that sort of went out of fashion a little bit, at least in predictive coding, but we're kind of working on similar versions now with hybrid inference. So rather than just doing top-down generative model and iterative feedback prediction errors going upwards, you can also have amortized inference, where you have a separate pathway which takes information or takes data and maps it directly onto posteriors. I wondered if that was interesting because it has some slightly different functionality, and that's a better description of what Hinton's model was doing. I think it's a separate feed-forward pathway.
Yeah, interesting. No, what we took from that was really the the top-down generative processes, exercised without constraint. That was the interesting property, without the constraint of input. And that's our model of REM sleep, basically. So that during that interval, the cholinergic excitement, this really fits with Hasselmo's model of cholinergic regulation of what he called the feedback pathway, but it's really the feed-forward pathway, that in REM, we are exercising this excitatory control. It's generative in a simple sense.
Yeah. Cool. Andreas, one more question?
Yes, I wanted to say, I think this is very strong evidence, or at least it points to a great direction for active inference. But in the diagram that you draw, I would say active inference is working in the one direction, outward, but the other direction I would think of as passive inference. I would say there's got to be a way, like where the environment affects us non-predictively. It's just simply, there's some faculty we have that it's simply telling us, right? And it's answering us. So active inference almost kind of implies that. But the fact that you divide it up into these two streams, I think that, would it be fair to say that there's, one half is active inference, but there's another half that's passive inference?
That's an interesting way to look at it. And what I would say, that gets us into the variational criticality domain. In other words, that the whole thing, I would say, is active inference. It requires both. You have to have the feed forward and the feedback. But sometimes the feedback must dominate. And I think animals show this. So a mouse, when it stops exploring, the theta turns off. So when the mouse is exploring, the theta is very strong, and the exploratory mode is really excitatory. But then it will stop and engage in consummatory, or we might even say reflective behavior. That's when we see the opposite. We see the external information as being integrated. So I think there are alternate periods of excitatory, exploratory, and more inhibitory understanding the environment, or allowing the sensory data to be predominant. But those are just phases of active inference. I wouldn't say the exploratory is only active inference. Yes, it's active, but it's the dialectical in a unique mode of excitatory inhibitory resonance that allows those two phases to become integrated.
That helps. And then with regard to the waves, what would be the role of the beta waves? Because the way I think about it, like gamma waves have the highest frequency, they'd be the lowest awareness. And then beta would be the second level. And then theta would be the longest, slowest frequency. That'd be the highest awareness.
Right.
So that's why I would have thought this would be a dialect between the beta and the gamma. But you're saying you see it between the theta. So I'm just wondering, what's the role of the beta?
The beta waves are really critical. And I have tried to, I couldn't integrate them in this talk here, but there's quite a bit we know about beta waves. They are fairly high level organization from the basal ganglia. And the basal ganglia loops through the subthalamic nucleus and back to the cortex. Those beta waves are inhibitory. They are this is the model of the status quo. When the beta waves go through the neocortex, they constrict or restrain the operation. They're clearly inhibitory, but in an active way, an active inhibitory maintenance of the status quo. And that's critical for stability of action. So, you have multiple inputs, but you need to maintain their stability over time. The beta oscillation and the kind of Pascal and Fries model provides that. and it's almost exclusively an inhibitory control over the same oscillations. So it's like a new layer of inhibitory control that reflects the basal ganglia's regulation of these
Mm-hmm.
cortical circuits. So it's like another overlay on top of this. And the interesting thing is it goes all the way down the neuraxis. This is a place where the higher level control, particularly from basal ganglia, fairly like executive operation of the forebrain, goes all the way down the brainstem into the spinal cord. And so the flexibility and stability have to be balanced, and beta is a key regulatory rhythm for that. So I like your effort to put together the different levels of oscillation. And I think you're right that beta has a very key role that wasn't in this talk.
And maybe to say in terms of a time scale, how that may play out, like I think of like before the 300 millisecond, line, you have unconscious answering, let's say, and then you have then you have this consciousness starting to figure out, okay, what to do with that, how to organize it. But I think that there's a break, that the full-fledged consciousness is holding a break and is basically preventing the conscious from prematurely imposing its model. It's saying, no, you have to wait. You have to. I've heard a talk about the play space, so to speak. Like you have to keep playing. It's a little bit like your REM dreams, right? Like, you have to keep going, and then you release the break. So if you think of like four levels of knowledge, whether, what, how, why, what is the answers? How is the conscious type of thing? Why holds the break? And so why, and see, so when you let go of the why, it becomes whether, which is the base, and then how becomes what. So that's the four levels of knowledge, how they would, this is just my metaphysics, but.
Well, no, I find that very interesting. And I think there's a neurophysiology underlying it. And I think you're right that beta has that kind of a break quality. So part of what Fawn and I have worked out, we call microgenesis. So we're interested in development, but the development of an idea. And that's essentially what you're talking about. There are stages in each consciousness or in each idea that have their own properties. And I fully agree that premature development of an idea is not necessarily good. Sometimes the break has to be released to allow the creative flexible stage, almost the REM stage of active inference, and before it becomes fully articulated. And there's a lot of classic psychology that looks at this exact micro developmental process, kind of in the way that you're suggesting, I think.
So I'll try to connect with you, but just to conclude, but I think it's a huge problem in consciousness studies that there's this fascination with qualia, conscious experience, what does it mean to be conscious of an object? Whereas if you throw away all your experience and you throw away all your preconceptions, you end up in a highly cognizant world. You'd end up with much higher consciousness, like where you're very much imposing, and you get to choose between those two streams, the upstream or the downstream. You get to see how they fit together. So I'll write to you about that. Thank you very much for your time.
Please do. No, I fully agree. And I think implicit memory is that space where you're not conscious, but you are integrating many elements that will eventually be very fruitful in consciousness. So I agree with you. Consciousness, I think, is an epiphenomenon in many ways. And then what the real work is the unconscious domains that we have minimal access to.
Thank you.
Okay, great. Perhaps on that note, we can hand over to Carl for some feedback. Thanks for your questions, Andreas.
Good. Is it my turn now?
Yes, it's your turn, Carl. All yours.
Thank you. Yeah, very much like Fan's talk. That was a wonderful survey. So many intriguing issues, and very much like last time, I think we've already had the best part, certainly in the presentation and in the discussion. And a little bit like fans talk, there's too much to talk about. So I'm going to focus. I'm going to focus on. Before I tell you what I'm going to focus on, I should say it was charming to hear about multiple sparse priors. I haven't heard about them for nearly a decade now, so I'm delighted that somebody's taking that technology forward. And indeed, in the context of electrical impedance mapping, did you know David Holder? Did you ever come across David Holder?
Yes,
Yeah.
yes, I know him well, and he's taught us a lot.
Brilliant. Yeah.
Yeah,
He was a vicarious friend of mine. He used to play in bands when we were students, with us, and then was a champion of EIT for many years. Anyway,
that's right, and made very important progress. Yeah.
but I can't talk about that and on another day, perhaps, but not in this meeting. What I'm going to do is trying to identify the cross-cutting themes. I know that we have worked together on many of these issues, but I'm going to pretend that we haven't, and stand back and try and spot the cross-cutting themes on offer here. And in particular, how to link them to the pure theoretical neurobiology and predictive coding of a sort of more technical sort. Because you clearly have, an eclectic and encyclopedic knowledge, not only the comparative anatomy, but also all of the your attendant physiological theories and consciousness theories, and you put it all together. The alternative perspective would be just to start from the maths and say what would inherit from this maths if something was sense making in the way that you've been talking about. I'm doing that because I think there are lots of really beautiful points of contact that you could celebrate, possibly in discussion and possibly in a series of different papers. So not so much developing the ideas, but more establishing their construct validity by saying, yes, this is exactly what the kind of conclusion you derive at if you just started from scratch, knowing nothing about anatomy, nothing about physiology. Well, a little bit about anatomy, a little bit about physiology, but just basing one's proposals or understanding of the computational architecture purely from the computation in and of itself. And I'm doing that because, as I say, there's some remarkable convergences which we've already sort of hinted at in terms of suggested references and the like. But for the current audience and people watching this recording, I'm just going to pretend that we haven't spoken about this and just give you my summary and feedback through that particular lens. Before I start going through the papers and just picking out on some sort of key graphics that speak to the many of the themes, not all of them, but many of the themes that you mentioned, I I'm looking at the cross-cutting theme here as feedback and feed-forward control of a excitatory and inhibitory nature being very, very specifically pitched at or considered in relation to the precision control, implicit in the encoding of uncertainty, implicit in the action of neuromodulatory transmitter systems, implicit in attention, thereby implicit in sleep and all other sort of perspectives. So I think that sort of one thing I think ties a lot of the arguments together, because you've covered many different themes, but the one thing which ties it all together, the particular and I'm going to suggest, to avoid confusion, and I know that we've discussed this and tried to convince reviewers and editors, I'm going to suggest not using feedback control, just backwards control and forwards control. I think that's going to simplify things for people like me or in the predictive coding world who are not familiar with this sort of tension between the sort of cybernetic feedback and the anatomical feedback. And it seems to me that when you talk about control, feedback, or sorry, backwards control and forwards control, I'm asking myself, what are you controlling? And I think the answer is you are controlling the excitability, the excitation inhibition balance, the precision. And that this has enormous implications for both microscopic and macroscopic computational architectures and our understanding of sense making in waking and in sleep, and also the deployment of attention in terms of endogenous versus exogenous attention that can be likened to the backwards control and the forwards control, respectively, that will have the colon. Well, sorry, I won't. You can unpack all the dialectics, with your expertise. But to me, that dialectic again, very much in the spirit of what we were talking about last week in terms of Leslie Anglad as what versus where. I think having that
Yeah.
dialectic underneath everything upon which people can hang various perspectives, I think will really help consolidate the story and so people enable people to say, "Oh, I see what he's talking about or what they're talking about." They're just looking at this fundamental dialectic from different perspectives, and the different kind of physiological, neurochemical, anatomical, functionalist attributes that this single dialectic offers as a frame. I say that at the beginning because the particular notion of resonance and NM phase coupling and the like, and particularly theta gamma phase coupling, does have an alternative explanation. So I'm just putting that out there from a purely maths point of view or sort of computational neuroscience point of view. There are two completely different stories you can tell about resonance and synchronization and coherence. One would be, I think, the story that you're more wedded to, and many people would be, which is the intimate relationship between coherence and the encoding of and greasing pathways and precision control in the sense we've been talking about it, coordinating, contextualizing bottom-up and top-down activity in the right kind of way to enable plasticity. There is another perspective on it, which I'll try to highlight with one of the papers, which has got nothing to do with that, which also may need to be nodded to, particularly in relation to hippocampal activity and place codes, sort of place cell notions. And I'll just spend a few moments just saying that there is another story here, so that people are clear in their mind what story you are telling. And I think the best story to pursue throughout this, it's all about the precision. It's all about the excitation inhibition balance. It's all about enabling the message passing in this sort of bidirectional fashion, in the right kind of way, contextualizing it, coordinating it, however you want to talk about it. So if you'll indulge me, I will now share my screen and font out. Good, that works. Yeah. Right. Sorry. In no particular order. Forgive me for this. And I appreciate that, we've been talking for some time, so I'm going to speak quite quickly. I just put this up just in relation to your last conversation about beta activity. There is in my world the notion that beta just is precision, such that when you need to engage sensory attenuation in order to move, you get beta suppression. And once you have moved and then you attend to the consequences of that movement, you have beta rebound. And that that story has been pursued in particular by people at Oxford University, but at UCL by people like James Kilner. So this paper came out about 5-6 years ago now, proposing that sensory motor beta power. So this does not pertain to the similar mechanisms you were talking about in relation to making a thought move or not having an idea, but this is actually moving. So sensory motor beta power reflects the precision weighting afforded to sensory prediction errors. It's a relatively straightforward story, and it speaks to something which you may want to allude to, which is the need for sensory attenuation to realize movement. If you don't ignore, sorry, let me just stand back. I'm assuming here that we all understand sensory precision as effectively the attention afforded to the sensorium. So if you attenuate your sensory precision, you are basically ignoring the sensorium, which of course you need to do in order to do sensory attenuation, to ignore the fact you're not moving. So if I had Parkinson's disease and wasn't able to ignore the fact I'm not moving, the the forward prediction errors, the feedback, I shouldn't use that. The forward prediction errors would just revise my prior beliefs that I am about to move, and I'm no longer going to sort of have a bradykinesia. But if I'm working properly, then I can contextualize through postsynaptic gain control, possibly in gamma motor neurons in the spinal cord, the precision afforded to the proprioceptive prediction errors that engage the reflexes, so that my prediction errors are then used to drive neuromuscular junctions to produce the predicted proprioceptive response, very much in the spirit of Feldman's equilibrium point hypothesis. But for me to move, I have to suspend my sensory precision temporarily, and then it will rebound again. And this, as I said, this has been pursued by Kilner and colleagues with a particular paradigm. And I thought, there's not going to be enough time to get into all the details, but if you had students who wanted to sort of join the dots at the level of the maths of predictive coding, then that maths is here unpacked in terms of the in this instance, the Kalman gain as a ratio of precision. So, so you can look at sort of precision control of the kind that you are focusing on as a really important aspect of optimal control theory and Bayesian filtering, which is getting the Kalman gain right, where you can look at the Kalman gain as a postsynaptic gain, as something that is set by neuromodulatory or at least voltage dependent, say NMDA receptors on usually GABAergic or inhibitory neurons. But mathematically, this is what it looks like, and then empirically, this is what you'd predict in terms of transient suppression and rebound of beta using EEG. So I thought that's a nice example of this generic story, that there's something about fast dynamics in the brain that reports precision, that you are now going to unpack in terms of cross-frequency coupling and theta gamma and communication through coherence. You mentioned Pascal Fries, and so that would be one. And of course, that inherits from Wolf Zinger, which is probably very much in the same spirit of as Yuri's Bizaka's work. So just for those people who want to go and forage later on, this is Palmer et al. In Brain, I think. Oh no, Neuroimage, 2019. Making a very
Now that's very relevant to our memory paradigm, because we will have actions, there'll be a response that goes. And so following that control process is very relevant to what we think of as the cognitive resonances of memory.
And indeed, that theme was exactly what I wanted to sort of unpack here. This comes from a particular paper, which I'll show you in a moment, but I think it was in one of the very first free energy principle papers, and I'm just making the point, which I think you should certainly at some point foreground, that in having a world model, in having a generative model, and having a forward model of your sensations that licenses this notion, this sort of inside-out notion of the brain as a constructive organ, creating explanations or predictions of the consequences of what's out there, but it doesn't know what's out there. That world model, that generative model, has three fundamental types of unknowns. And I think that's important for the current discussion, because we're saying that precision is where all the action is. It's attention, it's consciousness, it's mental action, it's coordination. It is the thing that makes us different from a thermostat or a virus. And it is interesting to just to think about, well, if I wrote down a generative model, what are my random variables? What are my own unknowns? And they come in three flavors. There's the states of affairs in the world causing my sensorium. There and they're sometimes referred to as hidden or latent states, simply speaking to this sort of inside-out perspective. Yeah, what's out there is never known. It's latent, it's hidden behind the sensory veil. Um, and then there's another set of unknowns, which is basically all the laws and contingencies, all the associations, the things that are reinforced in reinforcement learning, for example, and they become the parameters of a generative model. They're installed in the connectivity. And as you learn, that requires plasticity. And I think that is exactly the kind of learning that you were talking about, that it's not an episodic memory, or it could be if called upon in the right kind of way, but it's more obviously sort of like a procedural memory if it's in the motor domain, or an associative memory if it's purely in the perceptual domain. And that just basically says that you can understand sense making and inference in terms of from the point of view of predictive coding, minimizing prediction error, more generally in terms of self-evidencing, minimizing variation-free energy, as belief updating about states of the world through neural activity, and then in an exactly mathematical, symmetrical way, belief updating about the associations, the laws, the parameters, the connection strengths, which just is plasticity, and this is what learning is, and this is what memory rests upon. But the key point from your point of view or our point of view is there's a third set of unknowns, which is the uncertainty in play about both, and that itself has to be estimated. So you have to have very much like a statistician trying to make sense of experimental data. You've got to know your standard error in order to make any kind of inference. And the standard error is the inverse precision. It's the uncertainty, or the inverse uncertainty is the precision, or the confidence, or the negentropy. And that itself has to be estimated. Just making that, sort of graphically, this was making this fundamental point that it's not just about inference and learning. There's this third really important thing that has to be controlled, predicted, well, first of all, estimated and then controlled. If you've got sort of the capacity to infer the precision of everything at this point in time, then you have to actually enact this. And I think then you're licensed to use the word feed back and feed forward control, because you are now controlling the message passing, the neural dynamics that underwrites the belief updating, the inference, and the predictive coding at the same time as the memory formation through the plasticity and changes in the synaptic efficacy. I'll come back to this paper later on because it speaks to a range of things that you have brought to the table. So I won't get rid of that. I'll just start again at the beginning. It would be, I think, the final few sentences of these papers should always remind the reader that excitation inhibition balance is the primary explanatory vehicle for current understanding of many psychiatric disorders, and in particular psychiatry at the moment. So I'm just putting this up as a relatively recent review of excitation inhibition imbalance, and there are lots of people who speak to this, John Crystal, John Lisman, and lots and lots of other people think, getting very excited about this. And of course, from our perspective, this is a reflection of precision control. It's the relative excitability of say prediction error populations that are broadcasting in this forward sense the feedback to update to do your belief updating, not only about the contents of your percept, but also the belief updating of the parameters, which is the neuroplasticity that underwrites the learning. And this is my summary or review with about 200 references, just looking at this thing that is fundamental through the lens of predictive coding specifically, and with a throughout almost monothematic focus on precision, ranging from schizophrenia through to Parkinson's disease, with the requisite little diagrams, which I think you've actually shown during the presentation. I show this because I remember sort of trying to limit the number of references to 200 for space constraints, but there's lots of interesting references there showing how endemic this notion of precision is in computational psychiatry and neurology, for example. That was an interesting conversation about sort of the distinction between, first of all, the wake-sleep algorithm from Geoffrey Hinton and modern views of predictive coding, where the same sort of bottom-up and top-down aspect is exactly, I think, this sort of there's exactly this sort of joint forward and backward message passing on these hierarchies. It is a subtle point which I'm going to sort of half unpack or just, not unpack for, but sort of just to clarify what the discussion. So Will was absolutely right that the original introduction of the wake-sleep algorithm had these two separate networks, where there was one forward network used in the wrong way that sort of mapped from sensory input to sort of posterior beliefs or representation of what was going on, and then another network, which is for us the feed forward top down set of stream, which tried to predict from those representations what the sensory input was, and they were separately parameterized. Nowadays, and this was actually mentioned explicitly, that would be known as something called amortization, which effectively can be regarded as learning to infer. So how can you learn to infer? Well, if inference is done by predictive coding, then you have a very particular scheme, which most people now subscribe to in terms of understanding message passing in cortical hierarchies, that you've got these sort of ascending prediction errors and then the descending predictions, and they are self-organizing, and the prediction errors induce this belief updating as you ascend the hierarchy, and the predictions are necessary to generate the prediction errors. So there's only one network, but the ascending and descending parts are mutually in play in exactly the way, Don, that you were talking about. However, it is possible, if you observe the same kind of thing time and time and time again, you can now directly learn the mapping from the first sensory layer to the final representations, and basically short circuit or to amortize or to learn the inference implicit in the hierarchical message passing you get with hierarchical predictive coding. And that's called amortization, taking the pressure off all the reciprocal message passing and the coordination that we're interested in. And that brings us back to the original wake-sleep algorithm. So there is that distinction there. Whether you want to sort of unpack this for readers is another issue. It is a bit subtle, but it certainly is a big theme in machine learning and technical treatments of predictive coding. I think that what you're talking about is more along the lines of the way that Alan Hobson and I sort of loosely interpreted the wake-sleep algorithm as one, as noting that there were just two processes in play. There was a sort of prediction error ascending process to see how good you were doing, and a descending generative process of predictions that was necessary in order to generate the prediction errors, and then asking, well, what would happen if you now reduced the precision of the input, the sensory input? And the idea here is that very much like we're talking about sort of decompositions of expected free energy last time, that the log evidence, the log marginal likelihood, or the variational free energy, or the accumulated precision weighted prediction errors can always be expressed as accuracy minus complexity, or complexity minus accuracy. But if you've removed the accuracy by switching off, by attenuating through the sensory attenuation mechanism, through, say, a sort of selective cholinergic or adrenergic, or both, attenuation or sequestering from the sensorium, you just left, so there's no accuracy, you just left with the complexity. So you can now still maximize your model evidence through minimizing the complexity, doing the housekeeping, rehearsing stuff, checking whether something that was unpredicted during the day really needs to be remembered, or can you is that spurious and anomalous and redundant, and can you find a simpler model that explains everything else, for example. It was that basic philosophy of minimizing complexity in the absence of any sensory data that licensed a reference to the original wake-sleep algorithm, which used the same device. So in the sort of original wake-sleep algorithm, what you do is you expose during wake, you expose the ascending network to data. And then during sleep, you take away the data or assign it zero precision, and then let the descending one try to predict what was seen during the then let the descending one generate fictive data that then can be used for the ascending signals on the network to learn a simpler version of it. But the actual implementation in the context of predictive coding is much simpler. You just take away the precision of the sensory input and let it rehearse. And of course, how do you take away the precision of the sensory input? Well, in exactly the way that you've been describing, through neuromodulatory control, through the cholinergic and adrenergic systems that basically switch off the precision of the ascending prediction errors, so that you've got now the opportunity for learning during sleep those things that are self-generated, for example, during dreaming, in exactly the way that you were talking about. So this was written without any knowledge of, much knowledge of, sort of not your deep knowledge of the functional anatomy of the ascending neuromodulated transmitter systems. But I think again, there's this beautiful convergence between the importance of precision control and the kind of belief updating a brain would have in different modes in terms of being awake or sleep. And then we'll come to the difference between REM and non-REM in a second, which is interesting. So I should just say where this paper came from. This is a hypothesis, a white paper written primarily by Alan, with the technical bits supplemented by me where necessary. The interesting thing that I wanted to here we are. Yeah. Oh no, it's not. My apologies. Several years later, I'm just trying to sort of here, trying to speak to the distinction between REM and non-REM memory consolidation that you were talking about. And I'm trying to ask myself, would you have arrived at that kind of, again, it's a different kind of dialectic here, but it still rests upon selective precision control or sensory attenuation in this instance. Would you have got that from the maths? And I think the answer is yes. But it took a long time for the maths to reveal itself. And it revealed itself when we started to actually simulate sleep and look at the improvement one gets in terms of learning and rule discovery and model optimization. This is you don't need to even look at this or remember it, but it's just a graphic. It's comparing the emergence of the right kind of connectivity or parameterized generative model with and without sleep. and showing the improvement in terms of the efficiency, the sample efficiency, and the statistical efficiency of learning that underpins memory formation with sleep compared to without sleep. But the key point that this paper makes is there are two ways of doing that. And loosely speaking, I think you can associate them with your non-REM kind and the REM kind. Mathematically, there are two ways you can do it. You can either actively generate predictions and then try to recognize and classify and do predictive coding on the self-generated sensory input, which is replacing the actual attenuated sensory input. So all the way down, to some level, but not actually engaging the primary sensory cortex or possibly the sort of the colliculi. Sorry, the LGN. So you've got this sort of inside-out process during dreaming where you're generating fictive events, scenarios, and narratives. And the generative stuff is not countered by or constrained by prediction errors from the sensorium because they no longer have any precision. So you've got this totally uncontrolled hallucination, as Neil Seppo and Andy Clark might say. But the benefit of that is you can still learn during this self-generated sensorium. You can still learn. And what you learn, of course, is the simplest explanation for this self-generated stuff. And that basically removes redundant or spurious associations that you've learned during the day. Actively, by just rehearsing and learning online during this, it's not even an inside out, it's just an inside mode of perception that you have, you could associate with dreaming. But mathematically, there's another way of doing it, or simulating it. And that basically says, well, let's look at my connection strengths. And they record basically all the experiences and contingencies that I have experienced during the day. So all the information I need about my experiences during the previous day is actually installed in the patterns of connectivity. So what I can do is I can go back and ask, was this particular pattern the most efficient way of storing this information? And you can go back and just ask, well, if I'd started with this pattern of connectivity, would I have got a simpler model? Would my complexity have been minimized? So this is known as, in statistics, Bayesian model reduction. It's a kind of Bayesian model selection that does not rest upon any new data. It's exactly what you and I do after an experiment. We acquire our data, and then we stop acquiring data, and then we bring to those data a null and an alternative hypothesis and ask, what's got the most evidence in terms of my record of the data? And in these simulations, the record of the data is just the accumulated strength of the synaptic connections. So now I'm trying to put hypotheses to my connectivity and ask, what's the best explanation? And if I find the best one is removing redundant, reducing the model, I can get this synaptic regression of the kind that Giulio Tononi talks about. So to my mind, that sounds very much like your non-REM stuff. To my mind, it sounds as if this is, you don't need to actively create virtual inputs or fantasies or dreams. You can just do it without actually dreaming. Just by sort of putting. Obviously, these are not propositional hypotheses, but the maths is just like reduces to a very, very simple kind of synaptic regression. So I just put that out there as another way of saying, well, look, if you just think of the brain as trying to sort of having evolved in order to optimize the evidence for its models of the world, it is quite natural that it has evolved, despite the obvious sort of non-selective pressures for sleep, to sleep in order to do this kind of housekeeping, to do this kind of complexity minimization. And there are two ways of doing it. We can do it either by Bayesian model reduction, or we can do it by actually rehearsing and then learning the stuff that we're actually generating ourselves. So I thought that might be an interesting a little part of the story. There are lots of this is a paper curiously entitled "Active Inference: Curiosity and Insight," but it was largely to provide a vehicle to pursue a purely computational formulation of sleep from first principles using self-evidencing or free energy minimization. So that
That's very useful. That's a very nice way to summarize and to explain some of the paradoxical features, like the idea of a gist representation. I mean, how can that happen? I think the math kind of points to that gist efficiency that comes out of the process.
Well, that's very nice. And you should use exactly those words. I mean, certainly efficiency is a really important word because the complement of complexity is literally efficiency, statistical efficiency. So as you're maximizing your log evidence, you're minimizing complexity, you are literally maximizing your efficiency. And the other way, yes, I hadn't thought about that, but the notion of a gist just is getting that sort of bare bones, that sort of minimally but sufficient explanation of what's going on. It reminds me of Moshe Bar's notion of gist. Do you remember that when he was, so I'm pretty sure when he was in Boston, he used to talk a lot about, again, this was very much this sort of inside out view of perception, and he used to talk a lot about gist.
Yeah.
I remember him having little sort of diagrams. So again, that's another nice way to join the dots. Yeah, yeah. Um, okay, so where are we now? Yeah, I just now want to return to precision and briefly just run through a number of in technical endorsements of what you have said. The What's this about? Oh, yes. Precision viewed through the lens of a neuroanatomist um, who has spent most of his life working in the visual system, I think would be best summarized by the work of Stuart Shipp. I don't know if you know Stuart, but he's a sort
Oh,
of
yeah.
he was a student of Semizeki, and then was left alone when Semi retired. But he took very seriously the sort of the differential equations, the mechanics of predictive coding, and trying to understand it not from your large scale perspective, but much more from the point of view of the laminar message passing and the specialization of different cell types. And this was a relatively recent 2024 synthesis and review of this. And it is interesting that of all the things he could focus on, he focuses on precision control. He focuses on exactly the feed forward and feedback control that you're talking about. His particular interest, though, was more on the sort of microscopic, the microscopic yeah, so he just used backwards pathways with supposed to feedback, so that's quite good. But he spends a long time on precision and gain control and excitation inhibition balance implicitly. I haven't read this paper for a for a year, but I'm pretty sure that there will be some useful perspectives and references from this sort of more laminar mesoscopic analysis as opposed to the more macroscopic analysis that you're entertaining. So I thought, and it goes into gamma oscillations and the like as well. So I thought that might be an interesting paper to read if you haven't already read it, just because it sort of again speaks to this sort of cross-cutting theme of precision control. The other sort of way that cross-cutting theme emerges is in terms of your comments about the different neuromodulator transmitter systems. When one looks at the functional form of the generative models, again with no knowledge of their neuroanatomy or neurophysiology, one is forced to consider that because precision is an attribute of any Bayesian belief, any sort of mathematical belief structure, in the same sense that any distribution has a dispersion or an entropy or a variance. The same is true of any belief structure in the brain. And when one looks at the functional form of generative models, you get different, there are sort of three broad classes of belief structures or probability distributions or Bayesian beliefs that are implicitly encoded by either neural activity or synaptic efficacy. And they basically are the likelihood mapping from one level to the next level in a hierarchical context, or from the sensory input to the latent causes in a when you're starting off at the low end, at the bottom of any given hierarchy. That likelihood mapping basically is a mapping between levels of a hierarchical sort. And then within any level of the hierarchy, there are now mappings over time. So these are the dynamics of any state space models. It's, if I believe, I'm representing the state of the world, the latent states of the world as being at this course, at this sort of granular level, as being this now, what are they going to be in the future in order to predict in time, for example? So that's another kind of mapping, which is sometimes referred to as the dynamics mapping or the transition mapping or the transition probability matrix in a discrete context. And then there's finally, that's understanding how the world's evolving, but then it's what am I going to do? And then I have beliefs about, in say, the basal ganglia or in the extended motor system and planning system. I've now got beliefs about how my body is going to respond to this, and right through to what do I intend to do next. But those beliefs also have to have a precision. Most of the evidence that, again, this is written by a neurologist, and it's written about discrete state-space models as opposed to the continuous state-space models using predictive coding. But I think the basic story holds whether you're dealing with continuous or discrete state-space models. What was done here was basically associate these different kinds of precision with different neurotransmitter systems, which seem to be very conciliant with what you were talking about. So, for example, cholinergic transmitter systems from the nucleus basalis, these generally look as if they modulate the likelihood mappings that map to the hidden states. Whereas noradrenergic neurotransmission looks as if it modulates the precision of the dynamics of the transitions, so that if I'm very uncertain about what's going to happen next, I get very aroused, for example, or vice versa. Which is not quite the same as a sort of attending to this in the moment or that in the moment that you'll get more through a cholinergic neuromodulation. So again, if we talk about sort of things in terms of attention, we're either talking about attention to the future or uncertainty about the future, or we're talking about attention to or uncertainty about the sensorium or lower level constructs or representations. Both can have a distinct uncertainty. I can be very certain about what's going to happen next, even if the lights are off, and I have completely imprecise information. Or I can be completely uncertain about the future in this context, even though I can see everything very, very clearly with high precision. And of course, all of these precisions have to be estimated and carefully tuned in order to attend to the right thing. And then finally, of course, the dopamine, again, very consistent with your story, encodes the precision of beliefs about what I'm going to do next. So this is the planning as inference perspective on inference as inferring latent states, but in this instance, it's latent states that underwrite what my body's going to do next and what I'm going to do next. So I thought that was a nice way of mapping the neuroanatomy and the possibly even the neuropharmacology, but so the cell's origin of these really key neurotransmitter systems to precision, and to via the different kinds of precision beliefs that have the attribute of precision, their different functional roles in relation to the macroscopic anatomy that you were talking about, because you'd anticipate that dopamine was only really going to be in the game of modulating stuff that underwrites what I'm going to do. Whereas you might expect to find lots of cholinergic and adrenergic stuff, right, it's in the occipital or the auditory cortex. Not so much what I'm going to do, but just making sense of the world and doing sort of perceptual inference in this sort of coordinated way. Sorry, I should. And this is in Parr and Friston at the Royal Society, uncertainty, epistemics, and active inference. So you can just replace uncertainty with precision there, and then and if you're interested in the maths, if you actually wanted to simulate these kinds of things under a discrete state space models, the appendix actually has the free energy minimizing. So again, it's not simple, but it's straightforward maths. There is an optimal solution to the feedback and feed-forward control that in principle, you could actually use to simulate this kind of contextual guidance or modulation of excitation and inhibition balance in this instance. So that was that. This returns back to predictive coding, continuous state space models. You know about this work, but I think it's a nice example that says that exactly the same kind of phenomena. Well, to reinforce your point, the precision afforded the sensory evidence relative to your transition dynamics or your prior beliefs. So the prior beliefs are usually associated with the dynamics, the unfolding of latent states in time. If you don't have a nice balanced precision, then, for example, if you are too precise in your prior beliefs, you're basically effectively ignoring evidence at hand. On the other hand, if you're focusing just on the sensory inputs, then it could be you're attending to random fluctuations at the expense of not knowing your prior knowledge about the physics of this world or the way that this world works. So you have to get that balance right. And of course, that balance will look as if it's evincing generalized synchrony and critical slowing in exactly the way you were talking about. And this is a paper just basically showing that there is an intimate relationship, if not obligate relationship, when I am processing, in this example, simulated auditory input between the free energy um, um, and critical slowing and the criticality as measured by the Lyapunov exponents under sort of linear stability analysis. And these relationships are all revealed simply by getting the precision right. So the x-axis is just I think it's the prior precision in the figure. It was here, but finding the prior precision that matches the sensory precision or changing the sensory precision until it matches the prior precision. So from the point of view of a simple two-layer predictive coding architecture, it's just the precision of the prediction errors at the first level and the precision of the prediction errors at the second level, implementing your precision dynamics. But to my mind, this again speaks to this cross-cutting theme. It's all about precision control. It's all about that. And it's not just at one level, it's getting the balance right at all levels in order to enable this generalized synchrony and to enable this critical slowing and the requisite sensitivity. You know this paper because
Yeah.
It's,
Well, and this is key to, one of the new questions we have, which is we've tried to frame the criticality in these cortical mechanisms, but I think the broader implication, and it seems to fit with this analysis, is that criticality goes all the way down, that we need criticality of our arousal and precision modulation. These fundamental balances between feed forward and feedback levels or evidence versus priors. And that's a tricky business because now we're seeing that we have to have criticality of the brainstem and the midbrain. And we really have not thought about that before.
Yeah. You'd certainly need to have the brainstem and the midbrain carefully orchestrating the balance everywhere. I don't know whether you'd actually need the precision controller to be critical. I have to think about that. Right. Nearly finished now. The cross frequency coupling. I just put this up. I just noticed I'm a co-author. I didn't remember that I was, but I just wanted to, and I mentioned this gentleman to you before, who's the CEO of Neuroelectrics, but also a friend of Gustavo Deco in Barcelona in Spain. And he just has some interesting ideas about cross-frequency coupling as a neural substrate for prediction error evaluation. And what it turns out is that the evaluation he's talking about is exactly evaluating the precision or the uncertainty. So I think there's some convergence. I'm not sure at this stage that he's quite in a position to say, yes, that's exactly what I'm saying. But I imagine in a couple of years that there will be a sort of convergence on the importance of cross-frequency coupling in maintaining the right excitability and precision control in exactly the spirit that you were talking about in terms of the top down and the bottom up resonating in the right kind of way. So this is I think it's archived. I'm sure it would have been published by now, but if it hasn't, oh no, perhaps it's March 20. Anyway, if you want to speed read that, then see, it might be worthwhile reaching out to Julio. And I should just add just for everybody else, he with some other colleagues, including Gustavo and Francesca Castellano, are trying to set up a sort of academic institute for PhD students who want to study this kind of thing. So there's currently setting up a company to try and get funding to have workshops and possibly sort of PhD students and fellows to look at this kind of computational neuroscience that is deeply informed by neurophysiology and neuroanatomy. What's this about? Oh yes, this seems to be an important bit of empirical work which ties, which enables you to join the dots with communication through coherence and Pascal-Fries, which I think is exactly the same idea. It's basically when you're resonating, you get a more efficient message passing. That's just increasing the precision or the excitability or the coordination. And this work, talking explicitly about predictive coding models of laminar message passing. And the physiological, the spectral concomitants of that, using dynamic causal modeling in collaboration with Pascal-Fries using ECOG data. So if you don't know this work, it'll speak exactly to certain aspects of the importance of this cross-frequency coupling in terms of forward and backward message passing, where the messages now are carried at particular frequencies. So I thought that would be a useful nod to this work that was started by Andre Bastos, who you've mentioned. Would be an additional bit of endorsement or substance to the arguments. I can't remember the content of this paper, but I do remember writing a laudatory review of it. And I remember, because I can remember so this is my comment on somebody else's work, and I can't remember why, but I know that you need to go and read this other person's work, other group's work, and I know that because I remember thinking. Oh, yeah, perhaps it was well, anyway, it was a commentary on somebody else's work that had established these waves of prediction very much by looking at sort of the interaction between descending and ascending messages or dynamics in the visual hierarchy, and then the emergent traveling ways, very slow traveling ways, that might have indexed the phase locking or the NM phase relationships. And I was asked just to write a commentary upon it. It is just a comment. Anyway, yes, I'm sorry. You'll have to go and read that to find out why I was asked to write that. But it does refer to empirical work, which I thought was important. The final thing I wanted to do is just to come back to the yeah. We come back to this original figure and diagram, and what I've just remembered I haven't done is given you the alternative interpretation of theta gamma coupling, which I probably won't bother doing now. Just very briefly to say, when you have discrete state space models, it is the case that you have to do a lot of little discrete updates before getting the new bit of data. And this gives you a theta gamma coupling for free in the sense that every 250 milliseconds, I'm going to do little sort of 60 millisecond or gamma-like updates. And then after I've finished those updates and I've sort of made sense of everything in terms of not predictive coding, but belief propagation or variational message passing, which has a similar functional form, but is not it doesn't have the same architecture that you would get in a predictive coding scheme. So I do all my updates until, yes, I've now found the simplest, I found the best explanation on the basis of that data, and then I go and get my next data. So this is more consistent with a sort of embodied view of theta gamma coupling where I'm either whisking or I'm doing saccadic eye movements at a theta rhythm. So I'm getting data, I'm then shutting that down with sensory attenuation, and then I'm doing gamma updates in my head. And then when I've converged, I then go and get the next data. So I'm just saying there is another explanation for theta gamma coupling, which does not rely upon coordinated message passing and precision control. I'm just saying that's there. But now I want to return back to the first principles account of the symmetric, the frequency coupling and the predictions that you would get from a predictive coding scheme. Based upon this tripartite mean field approximation on the three kinds of things. And this was a very early, I think 2008, set of predictions that you would get if you committed to belief updating under these hierarchical dynamic models or hierarchical predictive coding models. One of the key ones that I think you've been speaking to is that the principal cells elaborating predictions, namely deep pyramidal cells, may show distinct low-pass dynamics, i.e. Lower frequency relative to those encoding the error, the feedback in our nomenclature, or the sort of the ascending prediction errors, say the superficial pyramidal cells. So talking about this spectral asymmetry, that has to be the case. And there's an interesting reason for it. Which actually only became there are lots of really interesting predictions here. Which you will recognize. Neuromodulatory factors play a dual role in modulating postsynaptic responsiveness and synaptic plasticity, which of course is the memory argument. You can go through these, but I just wanted to focus on that because there's a particularly interesting and really simple explanation for it that speaks to the NM phase coupling. If you have this inside-out view, and you are now thinking about the descending messages, the predictions being generated under a generative model, and then the prediction errors being reciprocated, ascending the hierarchies to do the belief updating or to adjust or to correct your expectations or your beliefs at each successive level, then that correction under a simple predictive coding or Bayesian prediction scheme is linear in the prediction errors. So those green arrows that you were showing from superficial to the ascending, in our terminology, the feedback part, they are linear driving effects because you just me as a high level representation is just moving to minimize my to move on to do a minimization through a gradient descent, and that gradient descent is driven by the prediction errors. So the dynamics, the forward dynamics, the ascending dynamics are linear, but of course our generative models are non-linear. They have occlusions, they have all sorts of interesting physics in them, which means that the predictions are non-linear in the activity, which means that you get things like period doubling. It means that if I send down a prediction, then the nonlinearities, basically like squaring a sine wave, induce a period doubling. So now you get this, you get, just because the generative model is non-linear, necessarily means you get cross-frequency coupling. That has to be synchronized when it's all working properly, in order for to minimize the prediction error or the free energy. So I thought that was a nice something that, if you like, just inherits. So notice this is not so much about this is the third story about the resonance aspect. I think you should focus on the precision control and the importance of coherence for greasing the pathways in the right kind of way and the implicit relationship with attention. But there is a simpler story just in terms of message passing, that just rests upon the nonlinearity. So as soon as you have a nonlinear generative model, you have cross-frequency coupling, but of a particular kind, when now you've got this cyclical dependency in terms of the differential equations, where the belief updating is driven linearly by prediction errors, but the belief generation is now generating nonlinear functions, nonlinear predictions that are forming the prediction errors. So I thought you'd like that. This
Very
was
definitely. That's an elegant thing that I would like to integrate that in this paper, because it really takes the first principles and puts them into an elegant framework.
excellent. That was it. That's all I have to say. That was hierarchical models in the brain and elsewhere. And that means I can stop sharing now. You've been very courteous and attentive. I've been wittering on now for nearly an hour. So thank you for that.
Well, this is all very relevant stuff. So I've been taking careful notes, and like fun, I'll be going back over the recording to be able to reconstruct it. So
What time is it where you are now? Is it time for breakfast yet, or?
yes. It's almost 8:30 a.m. So we're starting to get awake here.
You've got to admire the American work ethic, haven't you? Well, it's been a pleasure to see you. Thank you very much indeed.
This is great, Carl. Thanks very much. There's a lot to digest here that Fawn and I will spend some time now trying to put it together.
Brilliant.
Thank you very much, Don. Really enjoyed the presentation.
Great.
Thank you, Carl. Thanks everyone else for joining today.
Great. Thanks.
Great.
Bye-bye.
All the best. Have a great day.
Thank you. Bye-bye.